Most digestion and absorption of nutrients occurs in the long tubular structure called the small intestine. Its structure is specially adapted for these functions. Its length provides a large surface area for digestion and absorption. That area is further increased by circular folds, villi, and microvilli. The small intestine begins at the pyloric sphincter of the stomach, and then it coils through the central and inferior part of the abdominal cavity and eventually opens into the large intestine. It's usually about 10 feet long in a living person and about 20 feet long in a cadaver, because once you die, you lose that smooth muscle tone and contraction, and all of those folds are going to then be relaxed, and the length increases dramatically. So the small intestine is divided into 3 regions. The first part is the duodenum, or you may hear it called duodenum. I prefer duodenum because duodenum makes me think of 2 pair of jeans. So the duodenum is the shortest region. It starts at the pyloric sphincter of the stomach and is in the form of a C-shaped tube. The duodenum means 12. So it's named because it's about as long as the width of 12 fingers. The jejunum is the next part, and it's about 3 feet long and extends to the ileum. Jejunum means empty, which is how it's typically found after death. The final and longest part of the small intestine is the ileum, and it measures about 6 feet long and joins the large intestine at a smooth muscle sphincter called the ileocecal valve or ileocecal sphincter. The wall of the small intestine is composed of the same 4 layers that we've seen everywhere else in the GI tract: the mucosa, submucosa, muscularis, and the serosa. The epithelial layer of the small intestinal mucosa consists of simple columnar epithelial cells that contain many different types of cells. There are absorptive cells in the epithelium that are going to contain enzymes that digest food and possess lots and lots of microvilli that absorb nutrients in the small intestinal chyme. Goblet cells that secrete mucus, and intestinal cells that form crypts, or these deep little depressions that we're going to discuss in a few minutes. The submucosa contains intestinal or duodenal glands that secrete an alkaline mucus that helps to neutralize that gastric acid that was in the chyme. Even though the wall of the small intestine has the same 4 basic layers as the rest of the GI tract, special features are going to help to aid the process of digestion and absorption, and these include the circular folds, the villi, and the microvilli. Those circular folds are sometimes called the plicae circularis, and they're folds of the mucosa and the submucosa. They're near the proximal portion of the duodenum, and they end about the mid portion of the ileum. The circular folds enhance absorption by increasing surface area, so there's more for that nutrient-rich fluid to pass across, and it causes the chyme to spiral rather than moving in a straight line as it passes through the intestine. Also, we find on the small intestine villi, which are finger-like projections of the mucosa. There's a large number of villi, 20 to 40 per square millimeter, and this vastly increases the surface area of the epithelium available for absorption and digestion, and gives the intestinal mucosa a somewhat velvety appearance. They're embedded in the connective tissue, and within that we have an arteriole, a venule, and a capillary network, and a lacteal. That lacteal is a lymphatic capillary. So nutrients absorbed by the epithelial cells covering the villus pass through the wall of the capillary or a lacteal and then enter the blood or the lymph. And of course it's the lacteal that takes that fatty absorption to the blood, or into the lymph and then into the blood. In addition to these circular folds and the villi, the small intestine also has microvilli, which are projections of the apical membrane of the absorptive epithelial cells. When you view these through a microscope, the microvilli are too small for you to see them individually, so instead they just look like a fuzzy line, and we call that the brush border, and that extends into the lumen of the small intestine. It's estimated that there are 200 million microvilli per square millimeter in the small intestine, and this greatly enhances the surface area. So the larger area of this larger area allows for more digestive nutrients to diffuse into the absorptive cells. The brush border contains several enzymes that have digestive functions as well. So these brush border enzymes are carbohydrate digestive enzymes that are specific, such as maltase that breaks down maltose and sucrase that breaks down sucrose. There are protein digestive enzymes called peptidases and two types of nucleotide digesting enzymes. Those are nucleosides. There are also absorptive cells that can slough off into the lumen of the intestine, and as they break apart, they release enzymes that help to digest nutrients in the chyme. There are two types of movement that make up the mechanical digestion in the small intestine. Segmentations are localized mixing contractions that occur in portions of the intestine that have been distended by a large volume of chyme. These segmentations mix the chyme with digestive juices and bring all those particles of food into contact with the mucosa so that we can absorb it. They don't push the intestinal contents along the line. Segmentation breaks into segments. So it starts with the contractions of the circular muscle fibers in just a part of the small intestine, and that constricts the intestine into segments. Next, muscle fibers that circle the middle of each segment also contract, and that divides the segment again. So this sequence of events repeats. The chyme is sloshed back and forth. Segmentation occurs most rapidly in the duodenum, about 12 times a minute, and then slows to about 8 times per minute in the ileum. This movement is very similar to what you would get if you squeeze the middle and then the ends of a tube of toothpaste that has the cap still on. After most of the meal has been absorbed, the distension is lessened in the wall of the intestine and segmentation stops, and that's where peristalsis begins. So peristalsis is a propelling action that propels that chyme through the small intestine. This slowly migrates down the small intestine, reaching the end of the ileum in about 90 to 120 minutes after food reaches the small intestine. So let's recap where we're at with chemical digestion so far. In the mouth, salivary amylase converts starch into branched fragments of starch with 5 to 10 glucose units. In the stomach, pepsin converts proteins into smaller fragments of peptides, and lingual and gastric lipase converts some of the triglycerides into fatty acids and smaller glycerides, or diglycerides and monoglycerides. So when chyme enters the small intestine, it contains partially digested carbs, proteins, and lipids. The complete digestion of carbs, proteins, and lipids has to finish in the small intestine with the pancreatic juices and bile and the intestinal juice from the small intestine. So the brush border enzymes digest disaccharides into monosaccharides. That's small enough monosaccharides for the digestive system to absorb. Enzymes in the pancreatic juice such as trypsin, chymotrypsin, carboxypeptidase, and the brush border enzymes split any remaining peptides into single amino acids. Although some lipid digestion occurs in the stomach through the action of that lingual and gastric lipase, most of that digestion of lipids is going to occur in the small intestine through the action of the pancreatic lipase. So large lipid globules that have been broken down to smaller lipid globules by the emulsification of the bile provide a larger surface area, so the pancreatic lipase can function more effectively and break those remaining lipids down into fatty acids and glycerol that are small enough, like the amino acids and the monosaccharides, to be absorbed. Pancreatic juice contains nucleases, which digest RNA and DNA. These are going to further be digested by brush border enzymes that are down into small enough products to be absorbed. So all of the chemical and mechanical digestion from the mouth to the small intestines are directed toward changing food into forms that can pass through the absorptive epithelial cells that line the mucosa and then get into the underlying blood and lymphatic vessels. So monosaccharides, single amino acids, fatty acids, and glycerol. The passage of these nutrients from the gastrointestinal tract into the blood or lymph is called absorption. Absorption occurs through diffusion, facilitated diffusion, osmosis, and active transport. About 90% of all absorption of nutrients occurs in the small intestine. The other 10% or so occurs in the stomach and the large intestine. Any undigested or unabsorbable material that's left into the small intestine passes into the large intestine. Since we're looking at absorption, let's take a moment and look at a couple of factors related to that. The absorption of alcohol can, of course, directly determine the intoxicating effects of the alcohol based on blood alcohol level. Because alcohol is lipid soluble, it begins to be absorbed in the stomach. However, the surface area available for absorption is much greater in the small intestine than in the stomach. So when the alcohol passes into the duodenum, it's more easily absorbed. The longer the alcohol stays in the stomach, the more slowly alcohol blood levels rise. Because fatty acids and chymes slow gastric emptying, blood alcohol level will rise more slowly if the alcohol is consumed with very fatty foods like pizza, hamburgers, or nachos. While the enzyme alcohol dehydrogenase, which is present in the gastric mucosa cells, breaks down some of the alcohol to acetaldehyde, which is not intoxicating. So this slows that ability for the enzyme, the dehydrogenase, to digest or break down some of that alcohol when you have those fatty foods. Also, if you look at identical consumption of alcohol, females often develop a higher blood alcohol level and, of course, as a result of that, have greater intoxication than males of comparable size because the activity of the dehydrogenase is up to 60% lower in females than males. When you see mal in front of a word, it typically refers to bad. So a number of disorders where nutrients from the food are not properly absorbed are classified as malabsorption. It can be due to disorders that result in inadequate breakdown of the food during the digestive process, inadequate digestive enzymes or juices, damage to the lining of the small intestine from surgery or infection or certain drugs and alcohol, or impairment of motility. Symptoms can include diarrhea, weight loss, vitamin deficiencies, and bone demineralization.